Live. All right. Hi, everyone. Welcome back to Sussex Visions. I'm Michi, and we are really excited today to host Fabio Cortesi on YouTube here. And he's going to talk about vision in coral reef fishes. And yeah, we're very excited to hear from him. He's actually normally at the University of Queensland. Now he's going to speak to us from Switzerland. And yeah, take it away, Fabio.
Thank you very much, Michi. So I'll share my screen. In that case, oops. All good? Can you see?
Yes.
All right, perfect. Okay, welcome everyone. As Michi said, today I'll be talking to you about coral reef fish vision. And I thought aptly to start with that, I put up an image of all coral reef fish, different eye shapes and morphologies. Turns out there's a few non-reef fish in there. A cichlid made it in there, and also a few deep sea fish. See if you can spot them. But yeah, so I would like to start my talk by actually acknowledging the traditional owners of the country where we usually work, the sea country. These are the Dingal and Gurumungu and the Thanil people. They are the traditional owners of Lizard Island, which is a small remote island up on the northern Great Barrier Reef. I'd also like to acknowledge the traditional owners of the land where we do a lot of our work at the University of Queensland, which is the broader Yungambe language region. There's a few traditional owner groups that share that land. And I'd like to pay my respects to their elders, past, present, and also emerging, and especially acknowledge their spiritual connection to land and to sea country. And I think this is a really important point to make when you study these animals in the wild, especially if you're from not from there, if you're coming in as a colonizing nation, for example. We don't really know much about these animals to begin with. We're trying to understand them, but the traditional owners have been living alongside them for millennia, and they really have the best understanding of what those animals do in the wild. And so we need to learn a lot more from them. Okay, so in Brisbane, we are a group of ragtag people of about 20-ish students, postdocs, and Karen Cheney and myself as bigger group leaders of the Marine Sensory Ecology Group. And our joint interest is to study how animals see the world. I should thank now Dr. Abby Shaughnessy first for a lot of the slides I'm using here, because she actually made them. As a PI, you don't have time to do them anymore yourself, as you well know. Okay, so we try to study animal vision, or more specifically marine animal vision, using kind of an eco-evo-devo approach. I mean, it's a big word, but we are trying to understand the ecology of these animals, how it evolved, and how the development contributes to the different visual systems we see. We do this mostly in marine fish and also in cephalopods. I'm not going to show you any of the cephalopod work today, but if you're interested, just contact me. There's some really cool stuff coming there. All right, so, and most of the time, really, we're stuck behind the screen, but people think this is what we do as marine biologists, and we do that for a couple of weeks a year, which is essentially going out there and, as I said before, trying and look at these animals and understand what they do in the wild. And that involves a lot of diving. There's me wearing a Nemo hat up there in the corner, catching some clownfish, a lot of filming, and a lot of trying to observe of what these animals do in their day-to-day lives. So that's the best part of it, really. Since this is a neuro talk, I'm going to hold this really short, but just for a quick recap, in terms of vertebrate eye, if you look at the fish eye, it's very similar to a terrestrial vertebrate eye. It's got a lens, which is usually round in fish. You have the vitreous, then at the back of the camera type eye, you have the retina, which has multiple neuronal layers. And the one at the back of the retina is essentially the photoreceptor layer, which is the one we're going to talk most about today. I know Tom's lab works a lot on the other layers, and we're trying to do that too now. But most of the knowledge we have at the moment about the reef fish is still on the photoreceptor layer. And in the photoreceptor layer, we essentially have the two types of photoreceptors, which are cones and rods. I say generally because there's a few fish that actually do kind of a mix between the two, but we're not going to talk in detail about them today. Inside the photoreceptors, we have the cell body, the inner segment, the outer segment. In the outer segment, we have opsin proteins, which are bound to a chromophore. And depending on how this chromophore is bound and what type of chromophore it is sensitive to different wavelengths of light. So by changing the way the chromophore is bound within the opsin protein pocket, we can change the spectral sensitivity, or animals, including us, vertebrates and invertebrates, can change the photoreceptor spectral sensitivity, and thus what kind of wavelengths of light an animal is sensitive to. Now, the vertebrate ancestor, so millions of years ago, is thought to already have had 5 different types or subtypes of opsin genes, which conferred sensitivities to different wavelengths of light. These include an LWS, a red wavelength sensitive one, a UV sensitive one, SWS1, a bluish sensitive one, SWS2, RH2 being green, blue to green sensitive, and then RH1, the rhodopsin, which is mostly used in dim light conditions. And there's been a lot of work done over the years looking at the evolution of these different opsin genes in a lot of vertebrate species. For example, in humans, we lost ancestrally the green and the blue opsin genes. However, we still use kind of RGB, and that's thanks to shifting the UV to bluer sensitivity, and thanks to the application of the LWS opsin genes, where one of the 2 duplicates was shifted back towards green. It's more kind of yellow sensitive, actually, so it shouldn't be RGB, it should be kind of RYB, I guess. All right, so these 5 types of opsin genes, or opsin visual proteins that then form pigments, they're expressed across the vertebrate phylogeny, quite conserved in different photoreceptor types. So we generally have a UV sensitive photoreceptor, a bluish sensitive one, a green one, and a red one. And all sorts of combinations of these exist in different vertebrate species. But the general makeup of the different retinal layers and the photoreceptor layer with the photoreceptors is kind of conserved, let's put it that way. And Tom tried to gather us from the vision community, vertebrate vision community, a couple of years back, and have us write a section on the specific organisms we're interested in, to have a look at whether we can kind of tease apart the commonalities and the difference in the photoreceptor or in the retinal layers. And if you're interested in that, head to this review, big special issue in Seminars in Cell and Developmental Biology. Essentially, what is interesting is that the retinal layers themselves is fairly conserved, but the wiring within the layers between the photoreceptors and the interneurons, the bipolar cells, down to the ganglion cells, might be quite different between species. And most of what we know is essentially derived from the fish on the right, from my point of view, the zebrafish. So essentially, 99% of our knowledge, 90% of our knowledge about fish vision is derived from zebrafish. And my group is trying to make a dent into this and try and study it in reef fish to see if it differs or whether it's the same there too. But because the underlying evolutionary history of these photoreceptors is the same, the common ancestor already had these types of photoreceptors, we this year proposed as a big community effort to essentially standardize the nomenclature for these photoreceptor types. And so instead of calling them red or L or M, green or M, S, whatsoever you can find in the literature, we've suggested to name them essentially from photoreceptor 0 down to photoreceptor 7 in this case. There might be a few more added in the future. But this should make it easier to compare between studies and between study species in the future. And as you can see, the last column has the typical opsin that's expressed in these photoreceptor types. But especially if you look at the reef fish literature that I'm going to show you in a second and our research, these don't always match. So it kind of looks like the photoreceptors are ancestrally defined, but the opsin protein can switch between them. The fish can kind of choose which one to use and which photoreceptor type. And this is just another graph from Tom. I'm going to stop with his work soon. Chichango's going to talk about my work, but he did this really nicely. He summarized the amount or the number of studies into fish retinas, into fish vision over the years from 1950 to the 2020s. And as you can see, the zebrafish bit on the right is really, really on the rise, whereas everything else is kind of on the fall. So we're trying to curve that fall and bring it back up, generating a bit more interest into visual systems outside of the zebrafish model system. So yeah, most of what we know is from the zebrafish, and we're trying to compare it to it. So there's the last one of Tom's contributions to my talk, which is their suggestion of what the retinal circuitry looks like in zebrafish. And also the zebrafish community actually has quite a good online presence, and it would be cool if we could do this for the general vision community, not just zebrafish and zebrafish related research. Albeit the zebrafish rock is what they used to use as their common handle. That looks like it's gonna finish up soon. So we might have to use a different media other than social media to keep in contact. YouTube might be one of the ways. Okay, but I'm going on tangentials. Okay, so coming back to vision, if we look at vision in fish, it's actually much more complicated than what we find in zebrafish. If we take an opsin approach where we look at the visual opsins themselves, together with Dr. Zuzana Musilova from Prague University, Charles University in Prague in the Czech Republic, we set out a few years ago now actually to screen essentially fish genomes and look how many opsin genes there are. And what we found is that there are a lot. So fish, teleost fish especially, have a lot of visual opsin genes. And their evolutionary history is very complex. So starting out with the 5 ancestral subtypes, we see a lot of duplications, deletions. We also see a lot of gene conversion happening. And this differs between lineages, between families, sometimes even between species. So if we look at a given fish, it is quite hard to say how many visual opsins it has and what kind of visual system it might be using. We really have to dig down and take a kind of from molecular over physiology to behavior approach. And this I just wanted to add quickly, they don't only have very, very many visual opsin genes, they have many opsin genes in general. So photoreception seems to be super important in teleost fish especially, but in fish in general. And if we look at just opsins, so light-sensitive proteins across all types of tissues, fish generally have about 25 of these. And then visual opsins specifically, they usually have around 7. So 7 is quite a lot because if you do RGB, you can see most of the color already. If you add a UV or a far red, you pretty much cover the whole spectrum. So why do you need 7 of these? Which has been essentially the long-standing question in my group, and which we're trying to address still to this day. So I have so many bloody visual opsin genes. Some of the reasons for having this many genes is that we see a strong correlation with the light environment specific fish occurring. So if a fish lives shallower and is exposed to the essentially full spectrum of light that we also see in terrestrial systems, they tend to have more opsin gene copies. The deeper they go in the water column, the more restricted the light environment becomes, and the less visual opsin genes they have, but the more of a certain type they might have. So for example, once we approach the depths of the deep sea, below 200 meters, we see a lot of species that have a lot of green opsins, where green and blue light essentially dominates, but they've lost their reds and they usually lose their UVs. And then once we go really, really deep, they usually only have rods left, or rods, rhodopsin and a conopsin, a green opsin. So water depth is a strong predictor of opsin gene diversity. The other one is the time of activity or the time of the day a fish species is active. Generally, we see a lot more conopsins that are active during the day in diurnal species, which kind of makes sense. So there's strong selection on reducing your opsin gene in the repertoire if you don't really use it in your daily life. The third one is trophic ecology. So generally, fish that feed on zooplankton that either reflects or absorbs UV light tend to be able to see in ultraviolet. This includes the zebrafish and the baddenlap, which has been studied a lot. But for example, species that feed more on a herbivorous diet, so feed more on algae, they tend to both express higher levels of the red opsin, but they also tend to have more copies of these genes. So it kind of seems like there's a strong correlation, or there is a correlation between whatever the feeding ecology of a species is and the opsin gene repertoires they possess. The fourth one is color signaling. So the visual system and generally also the opsin gene repertoire correlates with the colors that animals use. And so, for example, if we look at these two reef fish here, they seem like your standard, yellow job, reef fish, everyday reef fish you see everywhere. And the top one is kind of brownish gray. So nothing exciting with a white tail, tons of them out on the reef. But if you look at them through a UV sensitive lens, you actually can see quite different patterns and colors that they use. So all of these, or many of the smaller reef fish, are actually sensitive to ultraviolet light. They do often feed on zooplankton, but they also more often than not use ultraviolet as a means to signal between each other. So a lot of them use ultraviolet for communication as well. The fifth one is the phylogeny. So there's quite a bit of phylogenetic inertia in visual systems, especially in coral reefs, and that usually correlates also with the color of the animals. So it's kind of a chicken and egg kind of question, what came first? It's not that easy to distinguish. The sensory drive hypothesis is actually very hard to prove. But if we look across the fish phylogeny, reef fish phylogeny, in this case, we have damselfish, then we do see a strong phylogenetic signal and a strong signal for color as well that defines the visual systems of these animals. And the last but not least, plasticity is quite common in the visual systems of these fish. And so it's kind of a pick and choose scenario where depending on how the light environment, for example, shifts, the visual system of the fish might shift as well. So they might be able to adapt. So for example, on a coral reef in winter, the reef is dimmer, the light environment is dimmer, but it's very broad and very blue and transmits all wavelengths, whereas in summer, it's much more turbid. The light might be more intense, but it also causes much more algal growth. So there's a higher degree of turbidity, and the whole light environment shifts towards the green or red wavelengths of light. And fish, we've shown now quite a lot of times that at least some reef fish can adapt their vision to these changes. All right, there's also other changes in the light environment that might cause an adaptation in the visual systems, such as diurnal cycles, which I mentioned before, seasonality, and also differences between microhabitats on the reef. So whether you live in a cave or under the ledge of a coral, you might experience quite a different light environment to when you live out in the broad daylight. And here's a picture of me in Okinawa last week, where you're supposed to have 40 meters visibility all the time. And unfortunately, we hit a day where we had about 2. But as you can see, the little clownfish is still, well, you can't see it here, but it was still reacting to me quite well, maybe because I looked like it. But it actually tried to bite me in the finger. So even when it's very turbid, when the light environment is quite different, these animals are still able to see quite well. And one aspect here to remember is that for small fish, especially for small reef fish, the visual world is quite restricted. So a lot of what they do, a lot of the behaviors we see occur over a distance of maximum a couple of meters. Once you move further away, once you're more than 5 to 10 meters away, they won't be able to spot you. So that's not important for them. So they don't need 40-meter visibility to do what they have to do to survive usually. All right, so in terms of phenotypic plasticity, this has been tested quite a bit in my group over the years, both in terms of ontogenetic changes. When reef fish come back and settle on the reef, they usually change their visual system over depth, as I've shown before, and also by actually putting reef fish under different light environments in the laboratory and then looking at whether they can adapt their visual system. And we find evidence for both. So some species seem to be plastic and others seem not to be plastic. And so the way we go at this usually is that we're trying to go literally from molecule to behavior. It's been said a million times, I know, but we're literally trying to do that. So we're trying to go from gene expression, now also epigenetics, over physiology, imaging, and then end up at the behavior of the animals themselves. Vision is quite beautiful for this because you have this strong link between gene expression and visual behavior of the animal. But the link is more often inferred than actually shown one-to-one, and we try to do that. So I just want to show you a quick story about molecular and physiological adaptations. As I said before, on shallow coral reefs, generally in winter, we have a really broad light spectrum. It's really clear, the water. It's when you want to go diving, it's a bit colder, but the visibility is really good, especially if you're shallow. And then in summer, it shifts more towards this greenish, murky light environment with a lot of material in the water. And we looked at the visual systems of reef fish in winter and summer and compared them using bulk transcriptomics to see, in this case, whether their opsin gene expression can track these changes in light environment. And we did this for a lot of fish, but I'm just going to show you a sister species pair, because what I want to show and point out is that we sometimes see really big differences, even in very closely related species. So, yeah, we did a lot of this work up on Lizard Island, which is that island I mentioned to you before. So maybe important as well to mention is that in coral reef fish, especially smaller coral reef fish, we generally have this sort of retinal mosaic. So we have a single cone in the middle, and then we have four, I'm gonna dare and call them double cones, but they're essentially two single cones fused together structures surrounding the center single cone. And the center single cone usually expresses a shorter wavelength sensitive opsin protein, often UV, SWS1 sensitive, SWS1, which confers ultraviolet sensitivity, but sometimes also SWS2B, which confers violet sensitivities, and often they co-express the two. So it's a bit hard to say whether what the ancestral photoreceptor type is that co-expresses both opsin proteins, because the opsin is not a good marker here. We have to do single cell to determine that. And then the double cones, the fused two single cones, they usually express a long wavelength sensitive protein in one member and a shorter, a bluish one in the other. And it's often RH2 green and LWS red, but also it can be RH2A and RH2B, which is essentially green and blue. So we see all sorts of combinations. But this is kind of important because the change we see, the plastic change we see between light environments occurs in all photoreceptor types. And the first one I'm going to show you occurs in the double cone photoreceptors. So in this case, we compared two chromis species. These are your normal blue jobs you see everywhere on coral reefs in the world. Chromis viridis is ubiquitous. It's circumglobal. You literally see it on every reef. Atripectoralis is its sister species. And when you look at them in human visible light, unless you're an expert, you will struggle to distinguish them. Atripectoralis has a little dot here on the pectoral fin. But yeah, I mean, the dot is also kind of here, so it's kind of hard to see. If you use a UV camera, they look very different. So under UV, you can distinguish them. But if you use human visible light, very hard. So what we found is that if we look at the expression of opsin gene within these single cones and within the double cones, then blue is winter and orange is summer. We do see quite a difference in viridis. So in viridis in winter, the RH2B, which is kind of more blue sensitive, is upregulated, whereas RH2A is downregulated. So the green is kind of downregulated in winter, the blue is upregulated. Whereas in Atripectoralis, which is its sister species, nothing happens. So it looks like one of the species is plastic, the other one is not. And we can also show this plasticity in opsin gene expression on the physiological level. So if we do something like electroretinogram measurements, if you use ERG, we can actually look at the response of the whole retina to different wavelengths of light and see whether in summer and winter this differs and whether it differs between the two species. So to do this, Abby built a transportable ERG system that we took with us to the field. And grounding it was challenging, let's put it that way. But she did manage eventually to get it to work. And what she did is she measured the response of the retinal response to different wavelengths of light, looking at the maximum intensity response curve. So what she did is she essentially illuminated the eye with different intensities as a specific wavelength of light, and then looked at how the retina reacted and looked at what point the photoreceptors would start bleaching. So essentially what we were interested mostly in was the beta wave amplitude, which will give you the bipolar cell response. So she was looking at what level of light or what intensity of light at a specific wavelength the photoreceptors would start bleaching. And so when she did that with fish in winter and summer, with viridis and atripectoralis, essentially what she found, what we're interested in is this half-width number here. Essentially what she found, to put it simple, is that in viridis it shifted. So in viridis, she could, for the longer wavelengths, 520, 560, 540, 580, I'm only showing you 580 here for simplicity purposes, she saw a shift in sensitivity to that specific wavelength of light. And essentially, it was a shift towards being more sensitive to broader wavelengths of light. Whereas in atripectoralis, on the right here, there was no difference between summer and winter. So the difference we see in gene expression, in opsin gene expression at the molecular level using whole transcriptomics approaches, we also can show it at the physiological level, which I think is quite cool. All right, so this is more sensitive to the middle to longer wavelengths in summer than in winter, which corresponds to the shift in opsin gene expression as well. Whereas not much happened at atripectoralis. Now, why is that important for us? Well, if we're trying to preserve the reef and preserve specific species, it might be quite important to know which ones can adapt to changes and rapid changes in light environment and which ones cannot. Simply because we have to triage which ones we have to put effort in to preserve. All right, we can also do this not only on the physiological level, we can take it one step further. And to do this, I'll show you a recent study we did in clownfish. Now, the question is, why clownfish? Well, for one, they're super charismatic. Everyone knows them. I mean, my little niece and nephew are here, and I show them a clownfish, and they yell out Nemo, and they're like 2, 3 years old, so they already know about them. They're also very good indicator species. So they're generally stuck to their anemone they live with. They live in a symbiosis with. So when the light environment changes, they don't really have the ability to swim away or find a more suitable habitat for them. They're essentially stuck to the anemones, and unless the anemone takes off, which happens sometimes but not too often, they have to cope with whatever is thrown at them. So they're very good bioindicators. The anemones also bleach, as we see here on the right. And once the anemone bleaches and dies, generally the anemone fish die along with it. They're also sequential hermaphrodites. So the males, they live in a size hierarchical structure. The biggest one is the female. The second biggest one is the reproducing male. All the other ones are helpers. When the female disappears or dies, the biggest male, the reproductive male, turns into the next female. So that's very interesting for us because initially there was this idea that they might change vision when they change sex. We haven't actually found any support for this yet. The other thing is, in terms of color, they're quite interesting. So all those white bits on clownfish are actually highly UV reflective. So if we look at this under UV or human visible, we can see that this very orange clownfish turns into kind of a black and white blob, and the white shines highly in ultraviolet. And they use this UV, so we can see it here on a, if we look at the spectral reflectance as well, there's a lot of UV in their skin color. They use this UV part of the spectrum to talk to each other, essentially. They can make it darker and lighter, and that also indicates the status in the hierarchy they have. The darker in UV, the more dominant the fish, essentially. All right. We put a lot of effort into developing reverse genetic techniques in these fish as well. So a couple of years back, my group, together with Sensei Kinoshita in Japan, we were the first ones independently to actually manage to get CRISPR to work in this system, and then we did kind of a joint revision article on it. We also able to knock in genetic elements and genes into clownfish. It's not as straightforward as in zebrafish, so this is by no means trivial. And the reproductive cycle for these animals is the generation time is very long. Like we're talking a year to a year and a half to get one generation going. So, and they're very finicky. The females choose the males, and if they decide that they don't like them one day, the male is gone. So a lot of work to get stable lines, but it does work. So in this case, on the left, we knocked out tyrosinase, so we ended up with non-pigmented fish. In this case, we just show the embryos. And we also managed to make fish with green hearts by knocking in some GFP. Last and not least, we have really good genomes for these species, for quite a lot of clownfish species now. There's 28 species out there. I think about 20 have now high resolution genomes, which are assembled to chromosome level, often annotated on par with zebrafish or even better. So we know that clownfish generally have about 8 visual opsin genes, and they confer spectral sensitivities ranging from UV violet over blue, green to kind of orangey red. And maybe important here, in their double cones, in their two fused single cones, in the longer wavelength sensitive codes, they generally express an RH2A, which is greenish sensitive, so this one here in the middle, and an RH2B opsin gene, which is more blue sensitive. So they don't use much LWS unless we change the light environment, but we're not going to go there today. All right, so if we look at the clownfish phylogeny and at their genomes, we also see some diversity even within these closely related species. So some of the species actually have two what looks like functional RH2A copies, whereas others only have one, each polygon being one gene copy, essentially. And then they all have two UV genes. We're not quite sure why. We're working on that at the moment. So even within this small radiation of fish and closely related species, we already see variability in opsin genes. Okay, this is what I showed you before. So generally in their photoreceptors, in their long wavelength photoreceptors, they have RH2A and RH2B. And in their short wavelength photoreceptor, they often either express the UV sensitive SWS1 alone, or they co-express SWS1 with SWS2B, the violet sensitive one, to essentially shift the spectral sensitivity a bit longer. So because they do this co-expression and because we see this variability between species, we hypothesize that at least some of the species should be quite plastic in their vision. And to test that, and because they're stuck to the anemone, of course. So to test that, we use a sort of behavioral experiment that's inspired by an Ishihara type color discrimination test. So this is the color plates we get at the optometrist to see whether we're colorblind or not as humans. I am somewhere on the color blindness spectrum, so I can actually not see the number in here, but I think a lot of people can. So it's a test that works very well to see whether animals can discriminate between chromaticity of colorful stimuli. I think, is it a 4? Michi, I cannot see it. Is it a 4? Should be a 4.
Yes, maybe. I can see the 4.
Yes, very good. Okay, so we can do this with fish. We don't show them numbers. But what we do is we have them pick the odd dot out. So the fish learns that they need to pick the dot that looks different to all the distractor dots around it. And once they pick it, they get a food reward. And so we built this RGB UV LED display that is able to stimulate all of the photoreceptors in clownfish specifically. And then we trained the fish to pick at the odd dot out, and then we tested different color lines. We tested different intensities of color, saturations of colors across the spectrum, essentially, across this color space of these fish. So for example, in this case, we went from a red, a very clear red that is easily distinguishable, to a more pinkish color that then essentially runs into gray. And the fish had to pick the odd color out until they could not be able to see it anymore. And that tells us the discrimination ability of that specific color of the fish of that specific color. And the lower, we call this a delta S, the lower the difference between the distractors and the color is, the harder it is usually to see. And using psychophysical models, the threshold usually for discrimination is assumed to be a delta S of 1. This is just the way it's modeled. So if something has a delta S above 1, the fish should theoretically be able to see it. If it's below 1, it cannot. It is not quite as linear and beautiful as we make it sound, but it kind of works. Let's put it that way. And so the way we do this is we train the fish to pick the dot, and then, as I said, they get a food reward. And I'm showing you one here, which is super easy to see. It's like a bright blue spot. And of course, the fish can kind of do it. So they go, they pick it, and they get a food reward, which is super cute. It sounds, well, it's actually not that easy, that hard to train the fish. We can train pretty much every fish we've tried so far, except for Apagonids, they're a bit difficult. And the clownfish can learn this within the space of 10 days to 2 weeks. But then it's a lot of behavioral testing. So the first, say, 10 days might be fun, and then the next 6 months are painful. But anyway, beautiful fish, good work to do for a lot of the students. So what we did is we wanted to know whether these fish can adapt their visual system when the light environment changes. And so we did this experiment where we exposed fish to simulated natural light environments under different conditions. So we had a UV plus environment where we had UV up to red light in the tank. We had a UV minus environment where we essentially cut the UV light off. And then we had a green environment where we shifted everything towards greener wavelengths. And we also had a brightness control, which I didn't put in here, but should be in here. Because the green is generally dimmer than the other light environments. We also did a brightness control just to make sure it's not the brightness that is causing the shift, but the actual change in wavelength spectrum. And so what we did is we left the fish under these light environments. Initially, we only left them for 10 days and then checked opsin gene expression. And we already saw a really, really big shift within those 10 days. But as I said before, to test the behavior, we already only need about 10 days to 2 weeks to get them to perform at the criteria we want them at. So for the behavioral experiments, we have actually left them under the light environment for about a month. And so what we did is we took a bunch of fish, we put them into the 3 different light environments. We left them in there for a month, during which they were trained to pick the odd dot out. Then we tested them. Then we swapped them around between light environments, left them under it for a month, tested them again, swapped them around again, left them under it for a month, and then tested their color discrimination again. The color discrimination was tested under a bright UV plus environment, so they had full light environment when they were tested, and they were tested for different colors, including green, blue, red, UV red, I think, and purple. I'll show you in a second. Along with the fish, the behavioral fish, we ran molecular fish, which were essentially these fish run in the same tank, but in a separated compartment. And they were sampled at each switching point. And then their opsin gene expression was measured using qPCR. So we essentially had three time points for this. The first time we switched, the second time we switched, and then at the end. And if we just look at the opsin gene expression, what we find is this. So if we go from a UV plus to a UV minus to a green, we see that on the left, we have the UV sensitive SWS1 opsin expression. And on the right, we have the violet sensitive SWS2B opsin expression. We see that they go from sensitive to mostly UV in the broad spectrum light environment to something that shifts more towards green in the green light environment. So we see a strong shift from pure UV to something more towards longer wavelength sensitive in the green light environment. They did not change much in the double cone opsin, so only the single ones change in this species. And if we test their discrimination ability, when kept under these light environments for about a month, what we find is that in the green, if we keep them in the green environment for about a month and then test their ability to discriminate green colors, they get better at discriminating green. They do not get better at discriminating any of the other colors we tested. So there's no difference whether we test them on violet, UV, red. I think we did blue as well. So essentially, if you put these fish under a green light environment, their opsin expression shifts towards something that should provide better visual resolution, better discrimination ability in green. And we can also measure that using behavioral experiment, which is your one-to-one link from gene expression to behavior, which is pretty cool. We can model this. So if we model the co-expression of UV and blue genes, how are we going? Oh, yeah, I got 5 minutes left. So if we model the co-expression of UV and violet opsin, so SWS1 and SWS2B in the single cone photoreceptor, we see that under a green light environment, so this is green, ideally the fish would express about 50-ish to 70% of this SWS2B to get over the discrimination threshold of 1. If they're around here, they will still be able to see all the colors we tested. So we can also show this on the speckle sensitivity level. If we co-express SWS2B increasingly in the photoreceptor, we shift the spectral sensitivity of the photoreceptor here on the y-axis towards longer and longer wavelengths. At about 70-75%, we see this big jump. And when that big jump happens in spectral sensitivity, we lose the ability to discriminate a lot of other colors. We can see green, but a lot of other colors we cannot. So if we still want to see all the other colors and green, we should keep it around here. Yeah. So we want an expression of around 50, 60, 70% to see all the colors that we presented them under a green light environment. And essentially, this is what we find. So if we look at the fish that were kept in a UV plus environment, they express about 70, almost 80, 75% SWS1. Whereas once we put them to a green environment, they end up with this kind of 50-ish, 50 to 60% expression or co-expression with SWS2B. So it looks like they really shift the expression of the opsins, which then changes the photoreceptor spectral sensitivity, which then allows them to essentially distinguish still green under a green light environment. And I think that's where I'm stopping today. We recently, together with Justin, it was led by Justin Marshall, this initiative, we wrote a book called Color in Nature. I recommend you buy the book, not because I'm getting royalties, unfortunately, but because it's a really beautiful kind of coffee table style book with a lot of images that make it easy to explain color to folks that don't have a scientific background. So it's an easy to show around book with very, very understandable text. And then, yeah, thank you to my various funding agencies that have funded this work. And I'm ready for questions.
Thanks a lot. Really cool talk. I forgot to mention this before the talk to the audience, but if you have questions, put them in chat and then I can forward them to Fabio. That's option 1. Option 2 is once we're done with this part of the questions, I'm going to post the Zoom link into the chat, and then people can join and ask Fabio personally if they want to. So the first question, I already have one from Tom, actually. He asked, since you mentioned that you were looking into a sex-specific change in clownfish at the retina level when they transition, do you see such a sexual dimorphism in the retina in other fishes? Or would you expect this?
We haven't published it, so I'm not going to answer. No, we do. We do, but in very extreme cases. So we see it in some of the deep sea fish, where they have difference in photophores, and they also have difference in opsin gene expression in the eye, and actually in like yellow filtering pigment they use. That has been published. In the reef fish, we haven't seen it except when the fish are ovulating. Usually the female opsin gene expression is a bit different, but that's probably strongly driven by the hormones that are being released. So the opsins are linked to thyroid hormone pathway. And so once thyroid hormone increases, they also start expressing some of the genes a bit stronger, but there's no strong sexual dimorphism. So it's not similar. It's not like what we see in butterflies or something like that, for example. in reef fish, as far as we've looked. Yeah.
Okay, cool, excellent. Then second question from Pranav. Do you think this opsin plasticity will be a boon for these fish since habitat destruction is a massive issue? And is this plasticity ancestral? Any thoughts?
Yeah, very good question. So I think it will be very beneficial for them, especially because of the habitat destruction and because we see a lot more sedimentation events with bigger storms. One of the avenues that we're pursuing at the moment is to look at the cost of it, because obviously building eye structures, building photoreceptors is very energetically costly. And if you keep switching them back and forth all the time, you would expect that eventually that would incur a high survival cost for the animals. But we actually haven't tested that yet. So yeah, something to do for the future.
Yeah, that would actually be. I'm gonna chip in here with my own question. So do you know how adding and removing these different opsins, if that changes downstream processing, like how the cones and stuff connect to bipolar cells and what changes at that level?
We don't know yet. We are trying to get there to a little bit together with Tom. I guess we're leaning on you guys and your expertise. But I can tell you that the change itself, I didn't show that part because I didn't have enough time. But I can tell you that they actually change the length of the photoreceptor. So they're not adding more cones or less cones between light treatments. What they're doing is changing the length of the outer segment to add more opsin when they're in green, for example, or when they're in UV plus. So I assume that the circuitry underneath is going to be the same, but honestly, I don't know. I don't know.
Okay. Then next question from Nora. If I'm correct, some time ago you had a paper about multibank retinas in the deep sea fish. I don't know if you mentioned it, but does the amount of layers depend on the environment the fish lives in?
Yeah, so a lot of the deep sea fish and some of the nocturnal fish have rod-only retinas. So they have retinas that only use rod photoreceptors. And in quite a few of them, they start building multiple retinal banks. So instead of only having one layer of rods, they have multiple rod layers stacked on top of each other. I think the maximum that we found was 16, but there's some species that have over 20 of these. So the retina, and they use very, very long and thin rods. So the retina becomes so thick that you can actually, when you dissect the eye out, it's like a clunky piece of tissue that you can see by eye. So it usually correlates with dim light vision. On coral reefs, we only see it in two fish groups. One are holocentrids, so your soldier fish and squirrel fish, and they were ancestrally deep or semi-deep, let's put it like that way. So they colonized the reef coming up from deeper environments. We also see it in moray eels, interestingly. So eels are very nocturnally active, all eel species. And it seems to be very advantageous for moray eels to have these multi-bank retinas.
Okay.
The idea is that you catch more photons, the more photoreceptors you essentially have.
I have another, thanks. I have another question from Tom. If I understand right, the opsin expression switch only makes sense if there is co-expression in a single photoreceptor, right? If so, would one therefore not expect much change in species where co-expression is minimal or where the opsins are very spectrally similar, like in zebrafish? Do we need to fix the lighting in our facility?
Yes, you have to fix your lighting, is the short answer. And that is correct. So we see a lot of plasticity in species that co-express opsins when we pick them up from the natural light environment already. We unfortunately don't have, maybe actually, if together with the cichlid literature, we might have enough data to do a proper phylogenetic comparative analysis on this. The idea was to get there eventually. So we're still racking up the number of species we're looking at their transcriptomes for, and then because the plasticity experiments are not super trivial. So we have to get the fish in and then see whether we can switch them or not. But yeah, the short answer is that. And so you want to have a stable light environment that entices your photoreceptors stably without them shifting around all the time. Yes.
Okay. Good to know. Okay. I think that's it for questions. for those of you who want to chat to Fabio, here would be the link to the Zoom room. Actually, we have one more question. Oh, yeah. Pranav asked if you think this plasticity is reversible by
Yes,
depending
so
on season. Yeah.
yes, it is. So definitely the seasonal one. So this is flexible. We see developmental plasticity as well, which kind of sets your phenotype as an adult. And so we do see that as well in a lot of fish, actually. But this is flexible. We've shown that with that experiment too, with the clownfish, where we switch them back and forth between light environments. And they essentially, I should have mentioned that maybe in more detail. They change their ability to discriminate green depending on what light environment you have them under. So they're better under one environment. When you keep them under green, they're better at seeing different types of green. When you then switch them to a broad light environment, they lose the ability to see those greens. When you put them back under a green environment, they regain it. So it definitely switches back and forth. Yes.
Yeah. So the link is currently in the chat. So let's see if anybody joins. In the meantime, I would have another question, which is in those fish you mentioned earlier, those two different ones you said you find in every reef.
Yeah.
One of them changes their spectral sensitivity, you said, right? The other one doesn't. Do you see any behavioral changes in the other one where maybe they go to shallower water or different parts of the reef so that they still maintain a closer, like less change in their spectral environment rather than changing on the retinal level?
Yeah, to be honest, no, we don't. But we know that the one that doesn't change, so the one that stays fixed on the UV, uses a lot more UV patterns to communicate, like essentially, especially when they're sexually mature, which is in summer when it is murkier, they use a lot more UV contrasting patterns on their body to communicate. So it could be that they want to maintain this high UV visual system. Mind you, the ones that shift a little bit, they're still sensitive to UV light. So it's not like the difference is that huge, but it could be that that's what it's driving it. We don't see any clear, like they both feed up in the water column on zooplankton. So, and they kind of roost together even on the coral heads. So like they're intermixed a lot of times. So you can't really tell.
I see.
But maybe that comes back to my initial point. We just need to sit there longer and look at them.
Yeah, monitor them over the year. Okay, so then I think the link's been out for a while. I'm gonna end the stream now. Thanks to everyone for tuning in. Let's keep this going. See you in probably a month for the next one. Bye.
Perfect. Thank you.
Okay, we are offline.
Yeah, all right.
Well, that was cool.
Yeah.
A lot of new stuff.